Good afternoon and welcome to join this online event from the Science Innovation Union, Oxford. So today we have invited expertise on both vaccine research and infectious disease modeling to jointly discuss prospects of the pandemic control with possible vaccine interventions. So in our live stream today, we have connected to two speakers, Dr. Mike Whelan and Professor Deirdre Hollingsworth. Welcome to both. Today's conversation will be divided into three sections. The first section will be an interview with Mike, where we'll know more of the ongoing vaccine development of SARS-CoV-2. In the second section, we'll try to understand the transmission dynamics of infectious diseases by interviewing with Deirdre. And the last section will be a panel discussion combining the two areas, where questions will also be taken from the audience, so every one of you. Each section will span for around 15 to 20 minutes. So to raise your question, please type your question here at the Pigeonhole, either by scanning this QR code or visiting pigeonhole.at and enter the passcode COVID-SIU. You can submit questions anytime during this hour. And if you want your question to be specifically addressed by one of the speakers, please refer to his or her name in your question. Questions or comments irrelevant to our topic today could be removed. You can also upvote others' questions to show your interest. So that's enough of me talking, and let's start our interview with Mike. And this will be hosted by our another SIU committee member, Ava. So Ava, I'll hand over to you.
Yep, thanks, Celine. So I'll start with giving a brief introduction of Mike. Mike Whelan is a PhD immunologist who is currently working as a project leader for CEPI, which is Coalition for Epidemic Preparedness Innovations. His portfolio includes vaccine for MERS, Rift Valley fever, and more recently, COVID-19. CEPI funds the development of vaccines for neglected pathogens with epidemic potential from preclinical right through to licensure, partnering with both academic and commercial entities. Mike has over 20 years of industrial biotechnology experience and 8 years of academic experience. He has his own consultancy company and was formerly the R&D director at ICER, working on VLP-based universal influenza vaccines, and the head of research at OnlyVax Limited, working on whole cell vaccines for prostate cancer. Mike, would you like to give a background overview?
Well, I think you pretty much covered it. I've been hanging around the vaccine space for about 30 odd years, but I've been at CEPI for nearly the last 3 years, and CEPI is quite a special body. We were formed in response to the 2014-16 Ebola outbreak, where the world said, enough, this shouldn't happen again. And that's what we've been put in place for. And it's probably the most worthwhile thing I've ever done.
Yep, then I guess we can start. We are aware that there are different technology platforms of vaccines, such as DNA, mRNA, and protein. So what is the difference in terms of safety, efficacy, testing, and production?
It's a great question, and there's no right or wrong answer. There's lots of vaccine technologies. I mean, the simplest one goes back to Jenner's time, where it was really just an inactivated virus. He used cowpox instead of smallpox. Same idea. In all cases, what essentially you're doing is fooling the immune system into thinking you've had a disease when you haven't. So yes, there are lots of tried and tested ones, but even in COVID-19, we've had to use some of the newer ones as well. So they all vary. They're all variations on a theme, really. As I said, the simplest way forward is to inactivate the virus, and that's fine so long as you can do it safely, and people are doing that. Alternatively, if we think about where the proteins come from, well, ultimately they come from DNA. So we could have a DNA vaccine, but essentially, if you think about it, that sequence encodes for whatever the target protein antigen is. How do we make sure it gets into the cell? That's the difficulty with DNA vaccines. On the plus side, they're very, very cheap, and they're potentially massively scalable. So it's got one plus side. On the negative side, how do you get it in there? People tend to use electroporation, which is quite difficult. Then the next level is an RNA vaccine, and these are very new. People like Moderna, which I noticed you've mentioned later on, that's an RNA vaccine. The big problem for years with RNA vaccines has been stability. And now people have got these new LNP formulations which seem to make it last longer. So it's a viable opportunity. That doesn't need to get into the nucleus, it needs to get into the cytoplasm, so it's a little bit easier. It's never been scaled before. That's the difficulty. You can make a lot of it, but does it work? We don't know. And then the next stage is a protein vaccine. Make a recombinant vaccine, a protein, which the virus would have made, but you don't make the rest of it. You don't make the nasty stuff. And again, your immune system thinks it's seen it. So with a protein vaccine, then we need to supplement it with something that will switch the immune system on, and that's usually an adjuvant. And that's just something which is, it's been called a danger signal by Polly Matzinger. And that's quite a good way of thinking about it. So there's lots of different ways. So in answer to your question, it goes without saying we wouldn't do anything that wasn't safe. You don't take risks with these things. That's just a given. So we always start with safety testing. So to be honest, that part of the question you can almost put to one side. We just wouldn't do it if it wasn't safe. So everything before it goes into human has properly preclinically tested and properly checked out. In terms of efficacy, well, we don't know. The tried and tested route is probably the protein route. That's the way most people would do. It'll probably work. But are some of the others better? Could you use less vaccine and make it go further? We don't know. In terms of production, cost of goods is a very big problem. And it's not about companies making money. For COVID-19, all our developers are doing this at cost. Nobody's making money out of it. But equally so, it has to be viable. There are 7.8 billion people on the planet Earth. How do we get enough vaccine for everyone? So if it's ludicrously expensive, it's just not going to happen. So we have to balance up efficacy and the ability to make it at a reasonable price. So it's a very long answer to a very short question.
Thanks, Mike. It was very clear. And how much progress have global efforts made on developing the SARS-CoV-2 vaccines, such as like the number of active candidates, whether it is in exploratory or clinical stage, and what are the main bodies that is taking part in this vaccine production?
Yeah, we reckon, people have been saying in the papers it's about 100. We actually think it's 260. we know of 260 groups worldwide working on it. Most are in the exploratory and preclinical stages, so testing in animals before they go into humans. You could count on the fingers of one hand how many are in advanced clinical trials. But there are a whole load coming. Within SEPU, we've got 10 active COVID-19 projects. So the question really is how quickly can they be safely brought through clinical trial? So it's maybe worth spending a minute or two just thinking about how clinical trials work. And there are three main phases. Well, technically there are four, but three main phases to get to a licensure. Phase one is a safety trial. You don't mess with a safety trial. We use a slow dose escalation and we make sure whatever we're doing is safe. Once that's established, then we can start to speed things up. Conventionally, you'd go to a phase 2 trial where you refine things, you get some way of measuring a response, you refine your dose, you refine your schedule, and you're just generally setting things up. And then a phase 3 trial is the registration trial. You do enough and show that this thing actually works, and you should get a license. Now, it's maybe worthwhile talking about licensure for a second. When CEPI was set up, as I said, we were really set up in response to Ebola. And you may or may not know, but the vaccine, there are now 2 licensed vaccines for it. The last one was only a few weeks ago. And neither of them went through the conventional phase 3 route. They got a license through emergency use. So actually, most of the vaccines were used under clinical trial protocol as a phase 2 product. So we know it's safe, we've done the safety trial, and we know it works. But we haven't taken it through the hundreds of thousands of people that you need to do to license it. And that's okay for something like an Ebola where we know it's going to be. It's not okay for COVID-19. We can't use a stockpile because it means everybody who got the vaccine would have to get it under a clinical trial protocol. And that's not going to work for the planet Earth. So we now need to license it. And this is something CEPI has not done before. So we're having to figure out ways in which we can really advance these clinical trials and get them done as fast as we can, maintaining safety, but get it to the point where we can license it and then everybody can have it. So this is why we're really trying to work at adaptive clinical designs of phase 2 and phase 3, where we sort of merge them together. And in fact, our colleagues in Oxford, I mean, that's been all over the news. That's exactly what they're doing. So they've shown it's safe, and then they're gradually building it out to get things moving as quickly as possible. And it's a pandemic. We've got to do things like that. We wouldn't normally do it like that, but that's how it works. So does that answer your question on that?
Yes. And one of the candidates that caught our attention is the mRNA-1273 being developed by Moderna. And as they are based in an emerging mRNA platform with potential advantages in efficacy, speed of development, and also the production scalability and reliability. And we noticed that this vaccine is actually based on previous studies on SARS and also MERS. Can you tell us more about that? Like, how is the target being chosen?
The target, well, first of all, let's have a word or two about Moderna. Moderna is one of the RNA platforms, so it's very new. They're very good. They've done some excellent work, and they've moved incredibly fast. We gave them some seed funding in January, and then they've actually been involved with Operation Warp Speed in America. So they've got huge amounts of money from the Americans, and good luck to them. It will move very quickly. The recent preclinical data looks very good. Their phase 1 data looks very good. We just don't know until we get it into a large-scale production how it's going to go. But it is an interesting technology. It may or may not work. It's never been tried, and there's no licensed vaccine based on it. So who knows? As I said earlier, it's got one very big plus in that you can make a lot of it very quickly and very cheaply. So if it works, it's a good one. Now, you asked about compared to SARS-1 and MERS. COVID-19 is actually conventionally called SARS-CoV-2. So it's SARS-2. It is very similar to SARS-1. It's much more glycosylated, and it's a bigger protein in the spike protein. The spike protein is the one that allows the virus to enter the cell. And it's pretty much every vaccine we've seen so far is based on spike. So if you can stop the spike attaching, you stop the infection. That's the logic. So in SARS-1, they did something very similar. And none of those vaccines progressed because the pandemic petered out much quicker than the current one we're in. We're now at a different ballgame. This isn't going to go away. This is going to be here for years unless we do something about it. So again, they've been able to use some of the learnings from SARS-1. A few of the antibodies cross-react, not terribly strongly, but they do. So we know we've got the right antigen. And Middle Eastern Respiratory Syndrome, very similar. We're still working on Middle Eastern Respiratory Syndrome. There are still outbreaks in the Middle East and most of the Saudi Republic, all around there. We're actively working on it. In fact, we've been working on it with our colleagues in Oxford, and they've done a clinical trial on it. So a lot of the technology that they used in their ChAdOx came from MERS. In fact, we funded their clinical trials. So we already know the platform was safe. Now, obviously, we should caveat that in that we didn't say just because you've used it once, you don't have to do a phase 1. We didn't say that. You have to do it. But we already had very good evidence that that platform was going to work. And Moderna have done something very similar. So it's based on previous experience of identifying what the antigen is likely to be, and then it's all about optimizing the platform. Okay.
We noticed that vaccines usually take years of clinical trials before releasing to the market, but the COVID-19 vaccines are progressing much more rapidly. How is this achieved?
Well, therein lies the rub. That's exactly what we're trying to do. And it's got to be a balance. We've got to do what's right for the world, but equally so, we've got to do what's right scientifically. So we can't take risks. Part of the way we've done it is we've only taken forward technologies which have been proven to work before and actually have shown that they can be scaled before. So there's lots of really good science we've actually had to leave on the shelf and say, yes, we'd love to do that, but not today, because it has to be quick. So we want to have the comfort, if you like, that these things can be scaled and can be made safely. So that's really where you would try and not cut corners, but try and speed things up. And then this comes back to what I said earlier on about the adaptive clinical trials. So the adaptive designs are such that you can move forward quicker. Once you've got the evidence you need, you can move to the next stage. This also has to be partly to do with the regulators. We work in a very, very heavily regulated industry, and quite rightly so. So the regulators need to be comfortable, but they are also changing the rules a little bit so we can move more rapidly. You know, we're getting proposals turned around from regulators within a day. That's never happened before. You know, so everybody's playing their part. But as I say, there's only so far you can go to speed things up, but you don't cut corners.
As you mentioned, there's like over 200 vaccine candidates under development. It will be important that all these efforts are coordinated. Can you tell us more on how is this achieved?
Yes, this is an interesting question. Coordination is very difficult. We work with our colleagues at WHO a lot, and they've come up with a number of things like clinical trial designs where they compare vaccines side by side. We have also been involved in something which we are pretty proud of, actually, that we call the ACT Accelerator, the Access to COVID Tools Accelerator. CEPI is a non-profit making foundation. We do not want to see people profiteering out of this, but we do want to help them get there and get things out as quickly as we can. So as part of the ACT Accelerator, there is CEPI, there is Gavi, the Global Alliance for Vaccines, and there's the WHO. And we will work together to prioritize those vaccines which are far enough in clinical development that they're ready to go. So it's not a question of saying one is better than the other. We're not trying to do that. We're just trying to get these things moved on in a coherent manner and in which it's balanced worldwide. One vaccine is not going to do it. We are going to need 3 or 4. And those are going to need to be scaled to incredible levels, levels which have never been seen before. So how quickly are we going to do it? And also, who gets it first? So we have seen what we call vaccine nationalism. We have seen countries say, right, we will give you X amount and we expect Y doses. I'm not going to name names here, but it has happened not a million miles away from here. But so it happens. Now, if we have, say we have funded the development of, say by this time next year, we have 500 million doses of vaccine, which is a hell of a lot. There are 7.8 billion people on the planet Earth. Who gets it first? So what we would like to do is have all of these go into the ACT accelerator, and then we'll have an independent body who will decide who gets it first. So we're thinking it's going to be at-risk individuals like healthcare workers, like the elderly in outbreak areas. And then we can build out from that. And that gets away from having governments say, right, it's our country and we don't care about the rest of the world. We don't want to do that. We think it should go where it's needed, regardless of the ability to pay. The finance of this is irrelevant.
So what are the main research and manufacturing challenges faced by vaccine developers?
Well, the biggest problem really is, how do we know it works? So we have got some unbelievably talented scientists we've been working with, and the whole scientific community has really stepped up, and it's quite humbling to work with these people. But what we've had to think about is we are starting from a baseline which is 0. COVID-19, SARS-2 did not exist until December last year. So there are no reagents, there's no animal models, there's no epidemiology, there are no clinical trials to compare. So how do we know what to do? So one of the other arms that we've funded is what we call enabling sciences. And we think this is very, very important. So this will include things like developing an animal model. There simply wasn't, in fact, there wasn't even the virus. I was in Australia meeting with one of our awardees in early February, and I actually managed to secure the first isolate of virus which had been taken by the Doherty Institute in Melbourne, and I managed to get it shipped back here to the UK. It's actually currently imported down, where they're able to do challenge models. That was the first one that had ever been isolated on the planet Earth. So we were involved in that. Then we have animal models being developed. There's all sorts of different ones, and we just don't know what works and what doesn't. Now, animal models is an emotive topic. Everybody, you know, yes, nobody likes it, but we have to do it, because if we don't, we risk taking risks with human lives, which can't be allowed. But we've had to develop models. Some of them worked, some of them didn't. I could give you a list, but it would probably bore you. But there's an awful lot. We've had to decide, as we've watched the epidemic, and as it's sort of hopefully slowing down for now in the UK, where would we do the clinical trial? So we need epidemiologists. So we need to know that if we do a trial in a certain area, we will get a certain number of cases. Because if we get that, then we can start to see an efficacy signal. If there's no cases, we won't know the vaccine works. Similarly, how are we going to know what we're looking for? Is it antibodies that neutralize the virus? Is it antibodies that can, say, complement fix? Is it a T-cell response? Nobody knows. It's probably virus neutralization, but nobody really knows. So we've had to fund some of those studies to start looking for correlates of protection. So there's a whole gamut of things that we need alongside the amazing research that we're working with. And everybody needs to fit in.
We know that people from different ethnic groups, such as African, Asian, could respond differently because their diversity in LHA, which is human leukocyte antigen type. Are clinical trials taking that into account? And how much do we know about this difference?
We don't know enough about it. It's a really good question, this one. This one has been bothering me for a while. By and large, people haven't really been doing it. I know from my own experience, I used to work in cancer vaccines, and cancer vaccines tend to be very MHC specific. Most people tend to go for something like HLA-A2, which is about 40% of Caucasians. We don't think that's right for a vaccine which is needed worldwide. COVID-19 doesn't discriminate. It doesn't care whether you're Caucasian or Asian or whatever. So we need a vaccine that covers everything. So we've tried to steer away from this. That said, the epidemiologists are certainly looking at it. And I think it's going to fit very nicely with what Deirdre is probably going to say, I would imagine, is big data. This is where big data comes in. We're going to have enormous data sets of full profiling, full immunological profiling of patients in numbers that we've never seen before. And it's when people that can handle big data can really drill into this, we will know the answer. Currently, we don't, but I'd love to find out. And I wouldn't be at all surprised if there were associations. There seems to be some sort of anecdotal evidence that perhaps it is different in different populations, but we just simply don't know yet.
And finally, I know that this is a hard question. When do you anticipate the vaccines to be available to the public?
Well, this comes back to what I was saying earlier on. I mean, we are pretty confident we are going to get a couple of vaccines. I mean, our friends in Oxford will tell you that they're going to vaccinate the entire population by the UK. That may or may not be true, we don't know. But we're certainly helping fund AstraZeneca to try and find out, and we'd love to see that. But that, with the best will in the world, it's not going to be possible to vaccinate the entire planet in 6 months. It's just not possible. And it's a question of, this comes back to the ACT Accelerator and why we think it's so important. If we can prioritize, that's going to buy us time to make enough vaccine for everyone. So we don't think that we should just be giving it away willy-nilly. It should be really targeted to where it's needed to try and damp down the response. And that's where the epidemiologists can come in and they can help us. Because at the moment, it's looking like it's going to have a degree of seasonality. So it does seem to be in the southern hemisphere at the moment as we head into summer. But it will come back. And we need to be ready, and we need to know where, and then we can target it. So yes, there will be vaccine available, you know, Q1, Q2 of next year. Is it going to be enough for the whole planet? No. So that's why we have to prioritize.
Thank you very much, Mike. Then, Eileen, I'll hand over to you.
Just to clarify that Eileen is also me. So Mike has already done me a favor in stressing out why we need epidemiologists. So we have one here today. So this vaccine stories all look very promising, but until vaccines become widely available, the only infection prevention approaches are like case isolation, contact tracing, social distancing, and hygiene measures. But to allocate the finite resources in the most effective way is crucial to understand the routes and timings for transmission. So in the next session, we'll get to know more about how the transmission is modeled and predicted. Before that, for those of you who joined later, I'd like to show how you can raise questions again. So if you go to pigeonhole.at and enter this passcode, you'll be able to raise your questions. And a brief introduction for Deirdre. So Deirdre Hollingsworth is a professor of infectious disease epidemiology at the Big Data Institute here at Oxford. Deirdre and her group uses mathematical models and statistical analysis to study the evolution and transmission dynamics of infectious diseases with the aim of informing the design of more effective control interventions. She leads the NTD Modeling Consortium, an international network of neglected tropical disease modelers. She has ongoing interest in the transmission and evolution of HIV, malaria, and influenza. She's also chair of the organizing committee for the Isaac Newton Institute's program on mathematical modeling of COVID-19. So we are really glad to have you here, Deirdre. First of all, can you give us a bit of background on the concept of infectious disease modeling, how people do that and why is that useful?
So infectious disease modeling is a way of rigorizing our thinking about how infections are spread. So we have some assumptions about how transmission occurs, and mathematics basically allows you to write those down in a rigorous way and then take them to their logical conclusion. And They're a tool amongst many others in the control of infectious diseases. They can be purely thought models, so it can be what if this, what would be the consequence of that, through to fully fitted, very detailed transmission models. And sometimes when we have really detailed models, so for example, that model the distribution of households in the UK, exactly who's interacting with who, sometimes it becomes very difficult to estimate statistical parameters or fit models to data. And so we often have something in between. So we have something that's less complex, but we can fit it to the data. And we really have a suite of models. And that's because, you know, infectious disease modeling is not the same as weather forecasting. We don't have experiments that will inform. We have some experiments, but we have, you know, limited data to inform our models. And so we tend to use them as a tool for thinking as much as a predictive statistical tool.
To know more about how to predict, and I prepared a very complicated question. Complicated enough that I want to share my screen.
Great.
So, especially relating to COVID-19, I've listed out a few factors that I can think of that could affect the transmission of this disease. So could you please spend some time to go through this list with us? For example, like for each of the factors, will they have an effect? Are they likely to be major or minor? And how much do we know about the estimates of these factors?
So this is the classic infectious disease modeling conversation. You need to understand what's your question. Why are you modeling the disease? So, and then you try and consider which of these factors are going to be important. So, as I was saying just now, like complexity is not always your friend in this situation. So if, for example, I was trying to understand the impact of contact tracing on transmission of the disease, not all of these factors would come into it. Okay, so but whereas if I was looking at the long-term patterns, for example, maybe the duration of immunity would start to become more important. And that's why we tend to adapt our models to answer particular questions, which can be frustrating, I think, for some people who are not familiar with our field, because you would think that you would build knowledge outwards and you would keep adding complexity, like all of these details, but actually you only use those details which are relevant for the question that you're asking. But you've made a fantastic list. Absolutely, these are very important issues. So when we think about how much people transmit to other people, this famous R rate, which you're hearing about in the news, it is driven by a number of these things. So that's why I've picked on it for the viral factors. So the longer the infectious period, the more people you may transmit to. But it does depend how infectious you are in that period. So that's something we're not exactly sure of. So the duration of the infectious period and how much of that can you cut off using measures like social distancing or quarantine. So for example, if people have symptoms and then they isolate themselves at that point, you reduce the infectious period. And that's why the proportion of infectiousness that's due to pre-symptomatic or asymptomatic, so people without symptoms, is really important. So there's your first one and your third one are tied together. The incubation period is also tied to that, because so incubation period is the time from when you're infected until you show symptoms. So that determines when you're likely to know that you're infected and when you're likely to isolate yourself. And again, if you're very infectious before that time, then it's very hard to use things like self-isolation to control the infectious disease. One of the important early signals that there was pre or asymptomatic transmission of this virus was the fact that the incubation period seemed to be about 5 days, although it can be as long as 14 days. And yet the doubling time of the epidemic seemed to be faster than that. So that's the rate at which new cases are emerging. And so one of the things I'll talk as I go through this is how do we gather evidence? So the incubation period we gather because, for example, before the lockdown, if we know that you were exposed to person A and we know when you were exposed to that person, then we can watch you to see when you show symptoms. And so you gather information from lots of people on how long it takes them to show symptoms and whether or not they show symptoms, and then we can learn about the incubation period. And that was one of the things that was actually known quite early on, because that's fairly simple to do as a study without any tests. So you don't need a PCR test, you don't need a serological test. Case fatality rate is a very important issue, but actually doesn't tend to affect transmission. So that's kind of downstream of the infection process. Now, seasonal variation, I was interested to hear what was just said about the signal for seasonal variation. So seasonal variation, the idea is that in the summer, for example, in the UK, we know we don't have as much influenza transmission. And this is thought to be a combination of both survival of the virus in the air and on surfaces, but also to do with our behaviour. So we don't tend to spend as much time indoors, for example. And there's a debate, ongoing debate, I would say, about the role of seasons in the transmission of coronavirus. And that's because for flu, the flu is actually kind of on a knife edge of almost not being an epidemic every year. It's kind of balancing on, the R rate for flu is kind of around 1.2, the estimates are. And so in the summer, we have that small reduction in transmission, and that's enough to push the R below 1. If the R for coronavirus is 3, for example, the estimates sit somewhere between like 2.5 and 5, then a little drop is not going to be enough to stop it transmitting in the summer. Now I'm going to stop going through because you asked a short question, even though you had a long list. So I'm going to take a breath and see if you want me to continue or if you want to redirect me to another question, Yusin.
Yeah, thank you very much for this very detailed answer. I'd like to follow up on a specific point that you mentioned about contact tracing.
Yes.
So how does manual contact tracing compare with automatic contact tracing? Are we going to have a phone app very soon?
So this is a really interesting and a very live question. So really the effectiveness of the digital contact tracing is entirely dependent on uptake and how many people use it. And I think one of the issues that they found in rolling it out in the trial in the Isle of Wight is that there hasn't been as much uptake as we would expect or hope perhaps to be effective. And the same issue is true of manual contact tracing. So will people report when they have symptoms? Will they be able to remember their contacts? How far back in time are you going to do? So one of the things about contact tracing, usually it's an attempt to do forward contact tracing. So try and identify the people that you are likely to have infected before they infect anybody else. But there's also the concept of backward contact tracing where you try and find who infected the person who's reporting to you. So if I had symptoms, I go to the contact tracer, I say to them, well, these are the people I've seen in the last week. And the contact tracer then tries to find how many of them are infected. But they might also try and find out who infected me and then check that they hadn't infected anybody as well. So, you know, there's lots of choices in contact tracing about how far back in time you look, which contacts you identify. So is it anybody you spent more than 15 minutes with? Is it anybody you spent 2 hours with? Is it people that you live with? And as we learn about this virus, we'll learn which of those people you're more likely to have infected. And those choices, those logistical issues are true both for the digital and the manual contact tracing. They're slightly different, but who you choose to activate and say you might be at risk is a choice. And that's why in most countries it's a combination of the manual and the digital contact tracing, because it's all about the efficacy of your contact tracing and the logistical challenges of your contact tracing. And it also depends on what the transmission rate is like in the community. Contact tracing, manual contact tracing, most of the models say, there's been a lot of papers, most of the models say it's a useful tool alongside others. It's not going to solve your problem, particularly manual contact tracing.
Yeah, I hope automatic tracing, if we really imply that we'll do a much better job, more label cost effectively than the manual contact tracing.
Yes, but there's even though there's been some really interesting papers about the ethics. You know, there are also reports where countries have decided to delete all the data and because there's been issues. So it's a complex ethical situation, and yeah, we'll see what the responses will be. Yeah, and that's why it's important to include such like an ethnic department within the Big Data Institute, right? Yeah, on my floor are really fantastic, and they wrote a really interesting paper around the digital contact tracing. Yeah, it came from the premise that if it's going to save lives, then that's your base from which you build a discussion around this kind of app. And so, yeah, it's really interesting for someone like me who's not an ethicist to understand those arguments.
Yes, exactly. So this is all very interesting. We can spend ages on this list, but let us get to a simpler question. So I'll stop sharing as well. So the next I'm interested about is, although we have huge progress made with rapid testing, like number of diagnoses, still not everyone who shows symptoms or at risk of close contact has been tested already. So on average, we see UK has roughly, say, 4,000 diagnosed cases per day. But how accurate do we currently know about what's the actual number of total cases in UK? And are we planning on having any results from large serological tests?
So very good question about how we measure it now. Well, there's two sources of information. One is the serological information, the studies, and the other is basically some inference around the rate of change of the number of cases and the number of cases reported, and then you try and infer how many cases are not being observed. And I think the models really differ a lot in that number, and I suspect that's why we're not seeing that number published, because it'll be very dependent on your assumptions about the proportion that are asymptomatic. There was a review published earlier this week, I think it was earlier this week, about a week ago, from which reviewed and said the proportion of asymptomatic infections was probably, I think they said 15 to 40%, which is a bit lower, I think, than a lot of people were thinking. And the WHO put out a statement saying that may be true. And I think there's still a lot of uncertainty around what asymptomatic really means. Because if I tell you that you're infected, you might be more likely to report symptoms than if you didn't know you were infected. And so there's always these problems with those types of studies. But you're right, also serological studies are hugely informative. And the one that came out a couple of weeks ago from the blood donors, which suggested that about 17% of blood donors in London had been previously infected, was really interesting and showed how different the London epidemic was from the rest of the UK. And I believe there are a number of ongoing efforts to do systematic sampling to understand the dynamics through the serology. And that's going to be hugely informative.
Yes, I'm looking forward to that as well. So going back to models, for models specific to COVID-19, I'm aware that the field is evolving rapidly, and we've seen several popular models, and you already mentioned one. So we also know that what factors need to be considered depends on what actually do you want to estimate. So among these models, can you just pick one that you find convincing and like very briefly introduce to us what are the main assumptions, methods, and what does it tell us?
So I'm going to avoid the question of plumping for one model, because we wouldn't be able to develop policy without multiple models. We need them. We need the parallel independent thinking to develop policy. So I'm not going to pick one. But basically, transmission is all about contact processes. And so you make some assumption about who's contacting who, and that can be data driven. So for example, we do surveys around, who did you speak to today? Did you know them before? Do you live with them? And that kind of thing. So there's been historically some contact surveys. They're not very big, it has to be said. There's a growing body of evidence. And then we have to have contact surveys after, for example, the lockdown to see how people's behavior changes. We have other models that make an assumption around information about household size. For example, the Office for National Statistics provides information on household size and age distributions in households. And then you make some assumption about how those households mix through schools and friendships and workplaces. So you can build up a network of contact patterns that way. So different ways of designing your contact network. And some more recent models are looking really at geographical small spaces and how people interact. But by small spaces, I mean small areas, statistics. So that's how you build up your contact network. So that's one set of assumptions. And then you have another set of assumptions around the progress of the disease. So as I was saying, this proportion of people who are asymptomatic, pre-symptomatic infectiousness, what do we understand about the life course of this disease? And the most important assumption I would say at the moment is how infectious are asymptomatic people. That's something we really don't know a lot about. And the other big unknown I would say about transmission is the role of children in transmission. What proportion? I think primary school children, I think we're quite clear that they are less likely to get infected and probably less likely to transmit onwards and less likely to have severe consequences. But older children, I think that the evidence is still quite mixed. So you can see how you build up a set of assumptions and you end up actually with a suite of models making different assumptions. And they're not necessarily nested, they may be quite different. And you need all those models to inform policy.
I think the next question actually connects really well with this one regarding the assumptions we need to make on like contact network or the percentage of pre or asymptomatic cases. So this question is, we see that the UK deaths due to COVID-19 has been dropping steadily since lockdown took effect. But we know the shops are now reopened now with distancing requirements. So is it possible to anticipate, will that cause a rise of the spread within 2 weeks time or more?
So in order to predict what's going to happen in 2 weeks time, we need to make some assumptions about changes in contact patterns. And there are data streams to inform that. So one of the data streams is mobile phone movement. How much are people moving around? You don't need the apps kind of identifying who's contacting who in terms of phones, but just where are people, where are the phones together? And that's really useful information. But the situation has changed now from before the lockdown. So if you and I were in a well-known supermarket together, our risk of transmission might be very different now to what it was before the lockdown. And so there's going to be challenges interpreting that change in movement in terms of a change in transmission. And there will be lots of people thinking about how do we measure that change in transmission indirectly and directly over the next couple of weeks.
Yeah. In order to produce more this kind of data, I may consider taking out my phone with me when I go out for running.
Yeah, exactly. Your phone is a very powerful data generator.
Exactly. So next question is a bit more relaxing, I guess. So with successful reduction in transmission seen in some areas or countries, it's probably time to start thinking about what might be an exit strategy looks like when all interventions could be lifted. So what do epidemiologists think regarding this?
There's a lot of debate. So we ran a workshop a couple of weeks ago and identified some modelling questions which are still open around what might be the exit strategy. The good thing is we're generating data all the time. So because every country is trying something different, I think that we will learn a lot. But it's important to remember that at the end of the day, it is a political decision. So the scientists may say, this is what you need to do to completely eliminate and be sure you've eliminated an infectious disease. But it's a political decision whether you want to take the economic hit or social. I mean, the social impact of the lockdown has been important. And the models can help guide that. But at the end of the day, we have to make a decision as a society about how we're going to balance those risks to particular parts of our population, our own individual risk, and the socio-economic impact of, really strict control measures.
Exactly. So the last question of this session might be a bit wild. So is there a pattern in human history showing that human beings will encounter this kind of global infectious diseases more and more often? Or in one word, will COVID-19 be the only battle?
No, definitely not. So I've only realized stupidly a few weeks into the epidemic that for me this was not a surprising event, and for other people it was. In SARS, it was a tragic outbreak, but it was controllable relatively quickly because anybody who showed symptoms was infectious, but only people who showed symptoms were infectious, pretty much. And so if you were able to isolate people who showed symptoms, the epidemic could be brought under control. Now that's not a small thing to do logistically, and it was, it took time, and sadly there were many deaths. But in terms of a global pandemic, that's actually a pretty good situation to be in. And then we had, bird flu and swine flu in particular in 2009, where we had an infection that looked like initially it had a high case fatality rate and definitely pre-symptomatic infection because it's flu. But as it turned out, the case fatality rate was not that bad. So then we were lucky again because we had a very infectious, rapidly spreading disease, but with pre-symptomatic and asymptomatic infectiousness, but fortunately not that high a case fatality rate. And so we always knew this was going to come, right? This combination of a high case fatality rate and pre and asymptomatic transmissibility. And yeah, I guess it was always going to come, and it probably will come again. And the difference now is how easy it is for people to travel around the world, and people have been writing commentaries and about this change in human behavior and how it's going to accelerate a global pandemic for many years. Again, it's not a surprise. I think the thing that was surprising to me was that we were able to just implement the lockdown, that it just happened and there wasn't any social challenge to it. There was an understanding that it was to protect the more vulnerable people in society. So that's the only thing that's really surprised me about this epidemic. I've been modelling social distancing interventions for years, but I didn't really know what that meant on the ground. And I was aware that it was a complex process, and it's surprising to see it have happened so well. And I hope we can find that balance between reducing transmission and reducing the socio-economic impact, but we'll see. I
guess we are all positively surprised in this end. Thank you very much. So in the next discussion, we prepared a few questions for both of you and maybe inspire some discussion. So Mike, may I invite you back to the screen? So first of these questions, generally, or COVID-19 specifically, how long can vaccine protection last? How does this length of protection compare with the immunity acquired from naturally being infected? And in terms of infectious disease models, is there a critical value of how long this protection needs to last?
Well, the simple answer is we haven't a clue.
Simple.
I mean, it's a new disease. We simply don't know. So all we can do, I mean, I think Deirdre summed it up really nicely when she said about, was it a surprise? No, it wasn't. We weren't surprised. She wasn't surprised. I mean, we've been waiting for this. We knew that this is why SEPI was set up, because we knew this was going to happen. But it's worth mentioning there was only one pandemic in the 21st century. This is our third. And they were all coronavirus. Well, with the exception of flu, the other two were coronaviruses. So was it a surprise? No, not really. How long the immunity lasts, we really don't know. With MERS, it does look as though it tends to drop off, although interestingly, you can still find antibodies years later because we've done it. But it does seem to drop off quite precipitously. Whether that's enough to give you protection, we simply don't know. And it could be the fact that there's about 200 coronaviruses and we only know about 10 of them. So, you know, there's potentially thousands of similar viruses. So whether or not you've got pre-existing immunity, we simply don't know. So are you going to require boosters? Possibly. We just simply don't know. And how long that lasts, how long is a piece of string? So we'll just have to, I mean, this is where we all have to work together. This is where the epidemiologists come in, the immunologists come in, and we just have to decide. And what's the correlate of protection? You know, it's all very well saying, oh, the antibody level drops off, but if it's not an antibody that's mediating protection, does it matter? We don't know. Unfortunately, that's a question you simply can't answer yet. We just keep modeling. Is that fair, Deirdre?
Yeah, absolutely. That's my understanding of the evidence base. We don't know how long immunity is going to last. We don't know if it's different for people who show symptoms and people who don't. We don't know how it's going to change with age. There's just so many questions. And is serology, as you were just saying, is serology the way that we can measure immunity in the population? We don't know. So this is, you know, it's those longer term dynamics that there really is the most uncertainty. And models are a useful tool for thinking about the what ifs. What if it looks like this? What if it looks like that? What does that mean for where we'll be in 6 months for different scenarios of lockdown and etc. You know, obviously if it's short-lived immunity, then we're likely to have more severe outbreaks. I think most people agree that it's likely to become what we would call an endemic virus. So we're going to be living with this virus for the foreseeable future. But which parts of the population are going to be at risk? Which parts of the population can we protect? How good is the vaccine going to be? Those are all challenges. But it's important to remember that that assumption sits underneath some of the decisions that some countries are making to try and completely eliminate the disease, or countries that are saying, well, okay, we can't eliminate this, but can we manage it? And so, because we can just wait for the vaccine and then we can kind of manage it from there. So there's interesting consequences of the fact that we know so little about the immunity. Initially, a lot of the models were assuming that there would be permanent immunity, but now the models are considering the fact that that may not be true, and that does change some of the advice.
So aside from the duration of vaccines, I imagine these models will also need to take into account like how much is the vaccine efficacy, what's information strategies, like what are the population to get it first, and also the time of being released, like if a vaccine is released now or in 6 months time, will that be similar effective? So is there any like good predictions on how does this different aspects of the vaccine affect the future course of this disease?
Well, part of that's going to be somewhat empirical as to who gets out first.
Mm-hmm.
And, you know, we definitely think there's going to be first and second generation vaccines. You know, and if the first generation vaccine is great, fantastic, but it might not be. But if it's enough to stop the pandemic and stop the spread, it's actually enough. And then you can come on with the second generation. So I don't think it's going to be as simple as that. It's not going to be a simple binary, this one has cured everything and that's the end of that. It's not going to work like that. We're going to have to be quite pragmatic in saying, yes, this works well enough to control an infection and stop the spread. And that's where we need to start. And then we can think about elimination. We're still trying to eliminate polio after 50 years.
Yeah, I think it's important to think about how a limited stock of vaccine would be used, as Mike was saying. You know, would it be used to protect healthcare workers, for example, seems like a likely use, or those who are particularly at risk, but often the people who are particularly at risk may not respond that well to a vaccine. So, you know, do you do a cocooning strategy where you, for example, vaccinate care workers who work in homes for the elderly? And it's important to remember in the UK as well, we don't always use vaccines just to protect those people at risk. The UK is unusual in that it vaccinates children for influenza, and that's because for influenza, unlike for coronavirus, as we understand so far, children are really the drivers of transmission of flu, and part of the reason we vaccinate children for influenza in the UK is not to protect them because they're less likely to have severe consequences, but to protect the rest of the community. So there are lots of different possible strategies depending on what the aim of your program is and what the profile of the vaccine is and how much vaccine you have. And so I'm sure all of these different scenarios are being investigated, and there will be time from when a vaccine there is some decision about that this vaccine is ready to be mass produced and rolled out for those strategies to be considered. And as you were saying, probably different waves of different vaccines.
Yes, exactly. So the actual number of people to be vaccinated, it varies between different aims we want to achieve. But if, say, in an ideal world, if we get a well enough vaccine, although not perfect, well enough means it's safe, efficient, can be manufactured massively, do we anticipate everyone to be voluntarily willing to take that vaccine? And how much do we of them need to be vaccinated or be infected and recovered in order to stop the transmission, like to imply the strategies?
You see, you're touching on two separate questions here, really. In an ideal world, what you're actually asking is how much do you need to get herd immunity? You know, and it's probably slightly lower than measles, you know, which is about in the 80–90%. You know, so it's probably a lot lower than that. But actually, practically, that's never going to happen because we're not going to have enough vaccine in a year's time. So it actually speaks much more to what Deirdre said about, do you use this cocooning strategy? And with what little you've got, how can you use it more effectively? Because we're just not going to reach that level for at least probably 2 years. So it's much more complicated that you can't stick those two questions together. It's not practically possible.
Mm-hmm. Mm.
Yeah, it's going to be a challenging situation, but every day that goes by, we have more information. We understand who's getting infected, who's transmitting. All of these things will play a part. Who's at risk of severe symptoms, who would respond well to a vaccine. You know, and if we want to protect, for example, cancer patients, is a vaccine going to be the right way? Would it be better to vaccinate people around them. And the UK is also unusual in that the national committee that decides on vaccinations has had mathematical modelers on it for many years. And so these you know, the Joint Committee on Vaccines and Immunisations, I believe it's what it's called. So the models will be in there, always with huge uncertainties, always uncertain with a new vaccine in terms of the impact on transmission. But yeah, we shall see. And I'm sure there's people trying to start that conversation in the public as well, trying to talk to us about what our expectations are from a vaccine and what would be the best use of it if we have a limited stock. So I would expect to see stories and comment pieces coming out as the vaccines start to look more likely.
Yeah. So now let's take a few questions from our audience. So the first one, most voted one, asked, after vaccinated, can one spread the virus? I think what they essentially means that you are testing antibodies or T cell that protect yourself, but can you protect others by not spreading it?
This is a great question, and I thought this might come up. I think somebody's been reading some papers recently, and 2 or 3 of the vaccine papers do look, and I'm not going to name names, but they do look as though the animals are definitely protected, but they're still shedding virus after a challenge. Now, we don't quite know why, and it doesn't seem to be prolonged. But for the first 3 or 4 days after the challenge, they're still secreting virus. And no, this is very uncertain. Now, it could be artifactual, this. So it could be related to the fact that it's an artificial challenge, and it's probably a lot higher than you would ever actually have in nature. So it could be that, but we really don't know. Certainly what we can say on the plus side is, I've now seen 3 or 4 published papers on new vaccines, and they are showing that the animals are not capable of being reinfected, which is great. But the question is, are they secreting live virus? And the answer is that they might be. But it doesn't seem to last. But for the first few days, it's definitely there.
And so an important question is it a lot less than they would have been had they not been vaccinated, for example, which I'm sure is included in those papers?
Yeah,
so,
yeah. And it mostly is, but how much is enough? That's the question.
Exactly.
And it also, as I say, it could even be an artifact of just that they've put in so much virus, you'd never actually see it anyway.
And that, you know, that ties to the 2-meter distance question as well, right? So how much virus is an infectious dose? That's something we still don't really know.
Here's another really interesting question. This Steve thinks much more than COVID-19. So he asked, will the need for COVID-19 vaccines manufacturer quickly take capacity away from other vaccine programs?
Ooh, we hope not. And we are actively trying to avoid that. I mean, the one it most likely impacts is the annual flu vaccines. And so currently we're in a sort of a lull position where they're not making the flu vaccine at the moment, so there is capacity available. But it's finding enough capacity for us to make the vaccine we need all the time, because as we head in towards the autumn, that capacity is going to be taken up by flu. And also, it would be morally wrong to take one disease away and replace it with another one. That would just be crazy. So it's really not a question of prioritizing one against the other, but it's a question of, have we got enough capacity to do what we need to do? But that's something we would never do.
Thank you. There's a very practical question. Some people is worried that have we opened non-essential business here in UK too early? For themselves, he asked, should I still self-isolate as much as possible, or can I resume some of my pre-COVID activities? I think
this is about personal risk, right? So clearly the fact that people are interacting more means there's a risk of transmission, a greater risk than there was a few weeks ago. But as I was saying, maybe those contacts are not the same. The transmission rate is not the same because people are socially distanced, they're washing their hands. You know, so even if people are going to the shops, are the risks of transmission amongst those people reduced? and how much is that? So, but the psychological effect of the lockdown and the social and economic impact of the lockdown has been huge, and those have huge health consequences as well. So as you as an individual making that choice, you need to decide what's the right thing for you in terms of your perceived risk outside. And so the understanding in the UK at the moment, the estimates of the reproduction number are that they are below 1. So it's likely there aren't many infectious people out there when you go out, but you still have to make that choice based on the information that you have. And if it's important for you to go out and go for a walk in the park or go to a shop to buy something just because you've been locked in the house for 12 weeks, that's an understandable choice that you may want to make. And there's no right or wrong answer to that. Individuals have to make their own choices. And just the thing that I try and do when I'm out, although it's hard to always be on the ball, is to try and act like you are infected. So try and act like you don't want to infect the people around you. And then I think that's a good guide for your behaviour. And then that protects you because you're unlikely to get infected if you behave in that way. So yeah, we're a vibrant society, and we need to sustain that. And it's been a difficult few months.
I think that's really important to hear as well, for us to all make our own decisions. So probably because of time, we'll take one last question from Adel. So he says, epidemic preparedness was forewarned. How can we sustain these levels of investment and collaborative efforts? Can this be a teachable moment for other diseases that affect the disadvantaged?
Great question, Adel. We're hoping this will be a turning point. CEPI, when we started, was a bit of a turning point, but now we're into a global area, in areas we never thought would happen. So we don't want this just to go away and be forgotten about. The large drug companies, the biotech companies, academia have all worked together incredibly over this. And to lose that impetus that we have now would just be awful. So we would like to keep that going. Yes, people have to make money at some point. But during the pandemic, everybody's saying this is non-profit. We've just got to do the right thing. And for the long-term future, building the infrastructure, now is the time to do it. Should we have been better prepared? Hindsight's 2020. But things like we're talking about building VMIC here in Oxfordshire, which will be the new manufacturing site. I mean, that's fantastic. That was actually on the cards already. It's being hurried up, but it was already on the cards. So that element of preparedness was there, but now it's the time to take it to the next level. And also having this sort of non-profit making area element for emergencies, that's the way it's got to go. And if we can sustain that, we think that's a wonderful thing. Deirdre, would you like to?
Yeah, I agree with everything you said, of course, and I think that, the SARS epidemic in 2003 was a real learning moment for the academic community. And I think, it had a long-term impact on the communities that were particularly affected. And clearly this is going to be a huge moment in our history and will be remembered for decades to come. And it will change a number of things in our society. And I'm not an expert in those things. But I think in terms of learning from other diseases, I work on a group of diseases called neglected tropical diseases, which are in the poorest communities in the globe. And they have a lot of attention from a lot of donors, and there have been millions of dollars spent. And it's really, they've had a huge impact on those diseases, but there's still a long way to go. And sustaining political interest in those kind of diseases is difficult. But once again, with coronavirus in the UK and in other places, it's the poorest populations which are most affected by this disease. It is people who don't have good living standards, people who are living very closely together. It's key workers that are not paid that well, who end up traveling by public transport to work and making large number of contacts at their work. We don't see those, we don't understand fully all those patterns yet, but the signals are that particular occupations and particular population groups have taken the burden of this disease. They've been the victims of this infection in the UK and elsewhere. And we have to learn that lesson. If we don't learn how to protect the health of those people, because they often have higher rates of the comorbidities, which make you more likely to have a severe disease. So I'm not an expert in those diseases either, but I just, you know, if we don't learn that lesson now and take action as we look forward over the next few months, but over the next years, we can't allow a particular population group to not only suffer everything else, but then suffer the pandemic worse. That's, you know, if you have to make the argument that it puts the richer people at risk, then fine. But, you know, we have to protect those people, and we can't allow that to continue. So sorry, that's a bit of a rant, but I kind of really upsets me that in a country like the UK, it's still the poorest populations that have borne the brunt of infection.
Exactly. I think that's a really important question and goes beyond the disease itself and goes through equity and all the questions we need to think about. So sadly, we need to leave it here because we are running out of time already. But we are really grateful to speakers today that can spare the time to share with us your views and insights. And also thank you for everyone who joined us. And sorry we can't answer other questions. But if you like this conversation, you can follow the publication by the Institute from our two speakers today. And you can also stay tuned for future SIU events. So thank you everyone again. Stay safe.
Thank you very much to the organisers, and very well done. Thank you.
Thanks a lot. Take care.
Thank you.